Audio will start shortly.
We'll talk about this in very nice detail. I only present this slide here because as someone who's been doing machine learning and deep learning, 160,000 neurons is extremely small. And this fly can do so much with so little. And so that's very interesting and exciting to me. So the degree of detail that we now have the connectivity for at this large scale is super exciting. And the question that I'm asking is, how do we turn these maps, these wiring diagrams, into simulations that can teach us about how the brain, how the fly brain actually works? So as a computational neuroscientist, here's my caricature of how I think the brain works. At the microscopic scale, it's a network of neurons. So of course, what matters is how this network is wired and connected, the neural connectivity. And then of course, what also matters is how these components, these neurons work. What are the biophysical properties of single neurons and synapses? And there's a host of other phenomena, including neuromodulation, plasticity, and maybe even more that we have yet to discover that comprise at the microscopic scale the substrate of the nervous system. And collectively, these give rise to the neural activity. That's a network phenomenon. And the neural activity gives rise to behavior. And the behavior that produced by this animal is usually in service of some task that it's set for itself, something that it's trying to accomplish. And super excitingly, we can now measure the connectivity in enormous detail. So you can ask, do we have enough information to build a simulation using this sort of bottom-up, these microscopic measurements that we have? The answer is no. Connectomes by themselves give you an incredible wealth of detail about the structure of the brain. These are measured in dead brains. And so they only tell you about the structure, but not about the dynamics in a living brain. And so, we have by themselves, they don't tell you for.
Audio will resume shortly.
The single neuron properties, or what are the synaptic properties for each individual synapse. So this is not enough. But good news is we have many other measurements that we can make from the brain. We can measure neural activity, not everywhere all the time, but we have very good techniques for measuring neural activity some of the time. We've been able to measure behavior for a long time, so that's very useful information. And in certain situations, you may know what the goal of the fly and might have been, especially if you've designed an experiment for this. So these other measurements could be used to constrain a single model of how the neural network in the brain computes. And so the talk here is about now that we have the connectome, but we don't know, we know the possibilities, the space of possibilities for single neuron and synaptic properties, but we don't know the exact parameters. But if we know something about the computation that collectively this network is trying to perform, the task, let's say, in what Tommy was talking about, it's maybe at the highest level of what the computational level is. Let's say we know that, and we know the details of the connectivity. Can we then put them together and now make predictions of neural activity at the level of single neurons? Can we now, with measurements at two different scales, make really nice predictions? So that's sort of the setup here. What we actually did is to build a simulation of the fly visual system. Many of you here know more about this than I do. What we did was through work at Janelia, as of a couple of years ago, we knew the connectivity was measured for about 64 cell types in the fly, in the motion pathways of the fly visual system. And so we took those measurements And we wanted to build a simulation using only measurements from a dead fly. So we took these measurements and then we wanted to build a simulation that then predicted neural activity measurements that were made by people here in the room and many others over 15, 20 years, 26 studies. And we asked, once we build this simulation without ever having fit this model to neural activity, can we predict the neural activity measurements made in the lab? And how well does our model make these predictions? If we can do this, then that suggests that this connectome is a really valuable measurement to have made now that we can make it super comprehensively. Okay, so you've heard a lot about the fly visual system. Here is a connection matrix at the level of cell types for 64 cell types in the motion pathways of the fruit fly visual system. And there's a couple of things to note here. Well, one, it's extremely sparsely connected. The connections only exist where there's squares, and the size of the square represents how strong those connections are on average between two neurons of those cell types. And there's also the sign, the color of the square tells you whether it's an excitatory connection or an inhibitory connection. And the way we know this is through the kinds of transcriptomic measurements of the neurotransmitter and the receptor that were made also from dead brains. So that's the sort of data that my colleagues at Janelia collected. In fact, that information that we have is not just at the level of cell types, but at the level of individual neurons. So if you take, and what we've done is put together a connectome for the entire right eye, about 45,000 neurons. This is what that connection matrix would look like if it's 45,000 by 45,000 grouped by cell types. So each little pixel here is one neuron. It's not a cell type, it's now a neuron. And we know something quite a bit about the spatial organization of the connectivity here, shown between MI9 and 2,4-D. So using these measurements, we can now put together a simulation that looks somewhat like this. The goal is to have to be able to predict the activities of individual neurons here. Here, I'm going to show you each hexagon is all the neurons of a particular cell type, and each pixel in this hexagon is one of those neurons. So it's a retinotopic map for all the neurons of that particular cell type, and the brightness there tells you how active that neuron is. So this is what we'd like to be able to predict is, let's say you put in some time-varying visual stimulus to the photoreceptors. What would the activity look like throughout the network as it propagates through the network? In order to predict that, of course, you need to know not just the connectivity, which tells you how many synapses there are, that's a structural measurement, but also the functional details of this. And so I'll just tell you very quickly, the only equations I'll show here, but maybe just look at the left side. What we decided to do here is to model the connectivity in great detail, but simplify the single neuron properties to the point where we just have passive point neurons. Many of these neurons don't spike, so that's a reasonable thing to do. So each neuron has two parameters. It has a time constant that says how quickly does it respond to input. And then it has a resting membrane potential, which says what is the voltage in the absence of input. These neurons are then coupled through synapses. These are graded release synapses that have essentially a threshold linear nonlinearity sitting at the presynaptic terminal that determines the amount of neurotransmitter that's released at a synapse as a function of the presynaptic voltage. And this is a threshold linear function. And so the synaptic inputs to this to a single neuron depends on the weights, who it's connected to and how strongly they're connected. And what we're doing here is we're saying that the weights are largely measured. using electron microscopy. This is what the connectome is telling us. And so the magic here is in how Wij, the weights between two neurons, is represented. And there's two components here. One component is the number of synapses between any pair of neurons that we measure. And the second component is a sign. This also we have a measurement for. So both of these things are measured. What we don't know from these measurements is, let's say there are 8 synapses between two neurons. How strong, how much current does a single one of those synapses carry? This is something we call the unitary synaptic strength. That's not something that we can get from a dead brain. So the simplest model that we can imagine building of this network still has 3 types of parameters that we don't have direct measurements for. It's the time constant, resting membrane potential, the single neuron properties, and then this alpha, which is the unitary, it's a synaptic property. What we hypothesized is that these parameters, whatever they are, probably have to do with the type of a neuron. So what proteins are these neurons making? And so all neurons of the same cell type will probably share these parameters, was our hypothesis. So we've tied them to the cell type. Similarly, for the unitary synaptic strength, it likely depends on what neurotransmitter was used and what receptor was used. And so you can just sort of say all synapses that have the same presynaptic neuron and postsynaptic neuron types have shared those parameters. So in this model, the number of parameters really scales with the number of cell types, but not the number of neurons. And so that's a massive reduction. And that's enabled by the fact that we actually measure most of what determines a synaptic weight through electron microscopy. So once we put that together, we still don't know what these values are. It's a small number of parameters, only about 700. If we didn't have the connectome, it would be about 2 million parameters, really. But with the connectome, it's a massive reduction, but we still need to figure out what those values are. And we don't have microscopic measurements of them. But we hypothesize that whatever those values are, they should be good for detecting motion. We know through much of the work here that the fly visual system, especially the motion pathways, are somewhere in there. Collectively, this network should be detecting motion. And so we asked, how might this network detect motion? And so what's the way that we operationalize that? We said, let's build a simulation and then show it video clips. In fact, video clips that were taken from computer vision, where for these video clips, we have knowledge of what the optic flow field might be. And this is a computer vision data set that was actually collected here in Tübingen by Michael Black's group. It's called Sintel. It was generated to train artificial neural networks to detect optic flow. And what is optic flow? Optic flow basically is a computer vision scientist's definition of motion, where if you have a video clip, for each frame in the video clip and for each pixel, you say, where does that pixel go to in the next frame? And so if you draw an arrow between where the pixel is now and where it will be in the next frame, that's a vector. And if you have those vectors for every pixel in the input, that's a vector field. And so these colors here are showing you the direction of that vector and the magnitude of that vector, how far that pixel is moving from frame to frame. And so we wanted to say that somewhere in this network, if this network is computing motion, it should be able to produce an optic flow field. But of course, the representation of motion used by the fly brain need not be the same as the representation of motion used by computer scientists in Tübingen. And so we have a motion decoder, which is a black box neural network that looks at the representation of a visual input that's generated by the fly visual system, and then tries to transform from that representation to the optic flow field. And so we train all of this end to end, and that's the way that we set those parameters, those 700 parameters that we didn't have direct measurements for. So now that was a way of intersecting, basically saying, what is a simulation that can A, detect motion, compute an optic flow field, and B, respect the connectivity measurements that we have through electron microscopy. If we put these two things together, what emerges? So if we put in all these bits over here, this process is called task optimization, popular now in neuroscience as well. Can we predict neural activity that's measured in the lab across multiple labs? So, the first thing we looked at was, can we replicate Oxford's finding in this paper of T4 neurons and T5 neurons being motion sensitive and in these cardinal directions. So this we get perfectly right. What I'm showing you is the motion selectivity of T4 neurons to bright edges on dark backgrounds, and T5 neurons to dark edges on bright backgrounds. And not only are these neurons direction selective, they achieve the right direction selectivity. In fact, you can look at the direction selectivity index for on edges, bright edges, and dark edges across this population of 64 cell types and ask, what does our model say? What does it predict across all these cell types? And we correctly predict that T4 neurons are motion selective, but its inputs are not. And similarly, T5 neurons are motion selective, and its inputs are not. And this is a recurrent neural network. This is actually a much more detailed, bigger network than what Oxford showed with just a few cell types. We're actually modeling everything from the photoreceptors to and many of these cell types have yet to be characterized even. So in some sense, these are all predictions that the model is now making. And all of these predictions, just to emphasize, came from a model that didn't know anything about these neural activity measurements. In fact, they came from a person who didn't know anything about these 50 years of research that you've heard about. And so, after we built this model, we've been going around and trying to figure out, what is known for this neuron, and are we predicting it correctly? And so, to me, that's a good sign. I think that we're not putting our thumb on the scale, and we're making these predictions. Of course, we also predicted the segregation into on and off pathways. So this is, we looked at 26 studies that, over 20 years. I think there's more studies that we're now still learning about and could probably add to the database, but a good fraction of this network is yet uncharacterized. But the cell types that have been characterized, even though they've been done in different labs, some with voltage, some with calcium, these sorts of measurements we're able to predict and predict them quite accurately. Now, what I've shown you here in these two plots that I've shown you are a bunch of circles for each cell type. So each of those circles, it turns out, instead of training with just one model, we've actually trained 50 models for each with the same connectome and on the same task. And so the idea is that you can think of each of these models as a hypothesis for what the neural activity in this network might look like and how this neural activity, how this network computes motion. So we're looking across these models to ask how robustly are these predictions made. And you can see that while individual models may make some predictions, might not get it correct, the bulk of the models the majority of them get them perfectly right. But we did ask, if we have 50 models, do they make 50 different predictions for the mechanism? Do they make 50 different predictions for the tuning? This is a coarse-grained prediction of the tuning, but if you look at the details, are they completely different? And so what we did was we took this model, we showed it a panel of natural video clips, and we asked, if I look at the neural activity pattern of a neuron in a particular model, in this case T4C neuron, and looked across models, so 50 models, and asked how similar are the neural activity patterns in response to the same stimulus, long sort of stimulus train. What we find is that they actually cluster into 3 clusters. So it's not like there's 50 solutions, there's only 3 solutions to T4C. And looking into these solutions, there's something interesting that we find. There's numbers associated with each of these clusters. The numbers are basically the performance on the optic flow test. That network that neuron came from, how good was that network at computing optic flow? Well, it turns out that the cluster with the best average task performance of computing optic flow has a direction selectivity prediction, and the direction is in the correct direction. The worst cluster is not direction selective, but the intermediate cluster is direction selective, but in the opposite direction. So this taught us 2 things. One, there seems to be a correlation between how well the network as a whole performs the task and the accuracy of the model's predictions. 2, we don't just get one solution, but we actually get a few solutions, but not like 50 solutions. And so that means that putting these measurements together and building a model like this gives us a small number of testable hypotheses that we can then dig into to try to figure out, what's going on. And so this is what we did. We looked at those 2 clusters, the top 2 clusters, which were both motion selective, and we looked at their inputs. And we asked, what's going on there? Why are these clusters both motion selective, but in the opposite directions? And what we found is that the cluster that gets the correct direction selectivity preference has the correct on and off, predicts the correct on and off response properties, the contrast preferences to the input neurons. But the incorrect cluster actually gets perfectly reverses the contrast preferences for these 2 inhibitory cell types, MI4 and MI9, which you heard of from Oxford. And so, even though we know the signs of all of these synapses, these inputs do receive, these neurons do receive inputs from, mixed signs. And so it's not a given that you would get these contrast preferences right. And so now what this means is, imagine that you were in a world where nobody had ever made any neural activity measurements. You had a connectome and you had this model. Now our model will tell us that there's these 2 clusters, 3, and if you make a measurement in just a T4 neuron, it also tells you what the correct activity patterns should be for these input neurons, for all of the input neurons. And so this is a way of really, exploiting all these measurements such that you only need a sparse number of measurements, activity measurements, in order to make very strong predictions about what all the other cell types in the system would be doing, which without the connectome, you wouldn't have a chance of doing that. Okay, so I'll just take a minute and summarize what we think we've shown you, and I hope what we've convinced you of, is that let's say you have 2 neural networks, and you've trained them to perform the same task. If those neurons, those networks, You also know something about the connectivity, the connectome. You've measured the connectome. And you build this model where the connectome comes from the measurements, and you've trained them both to perform the same task. Then the activities of the hidden units of this network are actually correlated. If you didn't have the connectomic measurements and you train two networks to perform the same task, you will never have any correlation between the hidden units. So you can't predict the hidden unit activity patterns. But with the connectome, you can predict that, and that means that not only can you predict activity, you can say something about the mechanism by which this network is performing a certain computation. So I hope I've shown you now that by combining these microscopic measurements with something macroscopic that's about the collective computation of the network, you can now predict neural activity in this network. And so where do we go next? Now, we live in this super exciting time where we have a connectome for the entire fly brain. So a naive sort of computational neuroscientist like me might say, what's next? Let's build a model of the entire nervous system. Can we do that? Well, we live in the age of AI and big models. And so can we build this? What would it take? So this is at this point where we're going to speculate a little bit. Let's say we're building a model of the whole brain. Let's call that a big model. Well, big models need big data. What big data can we bring to bear for building this model? Well, the connectome is not big in terms of terabytes, but we have it all. It's complete. And so once we have it, there's no more that you can collect. So we have all that. Neural activity will probably always be sparse and only in certain, conditions, specific experimental conditions. So it's small. So biophysics, again, that's small measurements. It's not really comprehensive. But behavior, we've been able to collect behavior for a long time. That's something that we can imagine just pointing a camera at flies and collecting huge amounts of that data. These are the data that we can imagine bringing to bear to build one model. And, how can we use behavior, animal behavior, to fit this model? So one of the missing links here is that you need a model of the body. The nervous system senses the world, and the way in which it senses the world is governed by the body of the animal. And the way in which it produces behavior is governed by the body, the actuators in the body, the limbs that it has, the degrees of freedom that it can move. And so if you think of the input-output function of the entire brain, the inputs are the sensory system, which can be specified in detail through a body model, and the outputs are the motor behaviors that are produced, which again can be specified in terms of the degrees of freedom, the limbs in this body. So this is what we set out to do. This sort of builds on work by our collaborators at Google DeepMind, who built this virtual rodent inside a physics simulator called Mujoko. And in parallel, Pavan Ramji's lab at EPFL has also built this anatomically detailed virtual fly model. But I'm going to tell you about the work that we did. And this, as I mentioned, is a collaboration involving a large number of folks at Janelia, including Gwyneth and our colleagues at Google DeepMind. So Igor, who you've heard of from Lisa's talk, who's a phenomenal anatomist, he imaged a fly, and what he did was then take that imaging data and constructed 3D meshes for each of the body parts. He put them together in Blender to build a model of the geometry of the body, which has 68 body parts that look like this, and 102 degrees of freedom. We then passed this off to our colleagues at Google DeepMind, who then brought it into the MuJoCo physics engine, which then allows us to bring this body to life by putting in actuators and putting in sensors into this model. And what's important in order to model behavior is not just the geometry and the physics of the body itself, but also how the body interacts with the world. Flies can fly. And so what that means is that we need to simulate how the body interacts with air in order to generate lift, drag, in order to be able to fly. So my colleagues, Yuval and his collaborators put together a phenomenological simulation of the fluid. And flies can also walk upside down and on walls. And so this is another thing that they extended this physics simulator to be able to model. And in my lab, we took that model and then we said, how do we then get it to generate the behaviors that real fruit flies can perform? In particular, we wanted to target locomotion, which is something that flies can do both in the air and on land. They can crawl on the ground as well as fly. And so we asked, rather than using the connectome at this point, just as a proof of concept, can we train an artificial neural network to do this? And this is indeed what we did using type of reinforcement learning called imitation learning, and just reinforcement learning, and it was a complete failure. And here you can see that if you just ask this body to produce walking gaits or fly along trajectories that were, in this case, measured in the lab, in Michael Dickinson's lab, it can do it, but in very unnaturalistic ways. What we did was we worked with data, for instance, for walking from Kristen Branson's lab, collected by Alice Roby, and then we trained this model to imitate the walking behavior of real flies. And then we're able to generate quite highly realistic looking walking patterns along arbitrary trajectories. And all these trajectories were were real trajectories generated by real flies in the lab. And the fly is asked to replicate that trajectory, and it can do so. Similarly, we did flight imitation learning for flight, and it's able to, these are trajectories in Michael Dickinson's lab where a fly was startled as it was flying along. Here's the startle, and at this point, it actually starts to fly sideways. This is really what it does. We get the position and orientation of the center of mass of the body exactly right. So now we have this body, which for fun, we also tried to do some visually guided behavior, like maintaining a certain height above the ground or flying through, navigating using its visual system through these trenches. All of these, we can now do in simulation. Although, I wouldn't say that the way in which these models are solving this task is necessarily realistic. So what we have now are good models of the body, the environment, and I hope soon we'll have the brain. These parts are in MuJoCo. This is open source, freely available. There's reinforcement learning as well that we have access to. And so I'd like to say that we now have the pieces, certainly the machine learning pieces that we might need in order to build a big model like this. And in particular, what we've developed are these red arrows. And the red arrows are machine learning methods for fitting a model to each of these types of data. And so with these in hand, I hope it's not too crazy to imagine building simulations like this. And with that, I'll thank you and take questions.